No, everybody's stunned into silence. So question, James, will RMLST identify new species?
So the species identification server will only identify things that already exist, but RMLST can be used for phylogenetic analysis. And so you can use, so you can build a phylogenetic tree with type strains of all the species, genus. And then if you have a genome of something that is you think is a new species, you could put that into that tree and see where it clusters. So that would give you some evidence to suggest whether or not, by looking at the tree and the branch lengths, whether or not that was a new species. But otherwise, it'll only show, like, the species identification server will only be able to tell you what's existing. So I guess the sort of follow on from that is that RMLST really think of it as having sort of these three different components: the sequence typing, the phylogenetic tree analysis, and the species identification server. And between the three different components, you'll be able to sort of answer the different questions that you have. It might not just be one of those. You might need two or all three. Definitely the phylogenetic tree analysis is very strong to be able to have all of those alleles predefined. And as I said in the talk, it only took 18 seconds to make that phylogenetic tree. That makes it a very very quick analysis to run and then rerun and add new isolates, and can continue to refine the question that you're asking. Makes it quite powerful based on the fact that it's so rapid.
Okay, that's great. Hans Christian is asking a question about how good RMLST is for species identification within the MITUS group. You can have a first go at that, and I can comment as well if you like. So Hans Christian's actually asking about the viridans streptococci and the MITUS group and differentiating different streptococcal species.
So I would, I deal with all species. So knowledge of very, one particular species or one particular group is not necessarily a fair question.
Okay, fair enough. So Hans Christian, we find it to be, go ahead.
But at the same time, what I would encourage you to do is try the server, try the website, try RMLST, draw phylogenetic trees, and see whether or not you get the resolution that you're looking for. I think you'll find that it has high resolution.
Yeah, Hans Christian, we use it quite a lot now in our with our genomes when we're looking at our own genomes or looking at other genomes that have been published, which are, for example, meant to be pneumococci but are contaminated with something else, are a mixture of pneumococci, or in fact are in the viridans streptococci group. So we find it incredibly useful, and I think it is quite good for picking up the other strep species. We certainly have used it quite a lot for that. Also for things like Pseudonemoniae, Streptococcus pseudonemoniae, and so on. Melissa will touch on this a little bit later in her talk as well, or more generally, not that specific question, but touch on how we've used that. So there's some other questions, James. So can it be used to identify species outside of MLST databases? I think maybe a comment on how many different species are actually in RMLST might answer that question.
So there's currently 12,000 different species in the database. Some of them are sampled extensively, and some only have one representative genome. But you can upload your genome to the server to see whether it gives you. It doesn't. So your genome doesn't have to be in the database in order to run an RMLST analysis. But RMLST really comes into its own when it's embedded within the isolate databases, like it is with Strep Pneumo, as Angela was saying. Because then you can store your allele designations, and then that makes it much faster.
Hannah, could you help, please? Thank you. So another question around contaminated genomes. The question is specifically, how will contaminated genomes be treated within RMLST? Maybe you could make a comment, James, more generally on what you might do with the genome if you identify it as being contaminated.
In the reference genome database, if I identify mixed samples, I remove them from the reference genome database because it messes up the power of the species identification server. If one entry has two genomes, but only one species annotation, then so I've tried as hard as I can to make sure that the reference genome library has only single genomes at each entry. But for other people, like in Strep Pneumo, it's up to the database, each database, how they handle the contaminated genomes within their own database. Quite often they put they found some contamination, they put that in the comments. Some just take them out completely, some leave them in because they're part of a larger study that's being published and they want to keep that for completeness.
Okay, I think we should stop there. We are running over time, I realize that, but I also recognize there are quite a few interesting questions in the chat that are helpful to address now where you have an opportunity to speak to the speakers themselves. So thank you very much, James.